Hello, my name is Joshua Mugabe. I welcome you to cell membrane. What is a membrane? Membrane is a delicate sheet that separates one region from another, blocking or permitting selectively or completely the passage of substances. The skin, for example, can be considered a membrane that separates the exterior from the interior of the body. Cellophane, used in the chemical laboratories to separate solutions, acts as a membrane too. Concerning their permeability, how are membranes classified? Membranes are classified as impermeable, permeable, semi-permeable, and selectively permeable. An impermeable membrane is that through which no substance passes. Semi-permeable membranes are those that let only solvent like water to pass through it. And permeable membranes are those that let solvent and solutes like ions and molecules to pass across it. They are also selectively permeable membrane. For example, membrane that, besides allowing the passage of solvent, let only some specific solute to pass while blocking others. What is diffusion? Diffusion is the spreading of a substance or molecules from regions where the substance is more concentrated to another region where it is less concentrated. For example, during boiling of water in the kitchen, gaseous water particles tend to uniformly spread in the air by diffusion. What is meant by concentration gradient? Is it correct to refer to concentration gradient of water? Concentration gradient is the difference of concentration of the substance between two regions. Concentration is the term used to designate the quantity of a solute divided by the total quantity of the solution. Since water is general, is the solvent in the situation, it is not correct to refer to concentration of water in a given solution. What is the difference between osmosis and diffusion? Osmosis is a phenomenon of the movement of solvent particles, in general water, from a region of lower solute concentration to a region of higher solute concentration. Whereas diffusion, on the other hand, is the movement of solutes from a region of higher solute concentration to a region of lower solute concentration. One can consider osmosis as the movement of water, solvent, and diffusion as the movement of solute, both concentration gradient driven. What is osmotic pressure? Osmotic pressure is the pressure created in an aqueous solution by a region of lower solute concentration upon a region of higher solute concentration, forcing the passage of water from that to this more concentrated region. The intensity of the osmotic pressure, that is in unit of pressure, is equal to the pressure that is necessary to apply in the solution to prevent its dilution by entering of water by osmosis. It is possible to apply in the solution another pressure in contrary way to the osmotic pressure, like the hydrostatic pressure of the liquid or the atmospheric pressure. In plant cells, for example, rigid cell wall makes the opposite pressure against the tendency of water to enter when the cell is put in a hypotonic environment. Microscopically, the pressure contrary to the osmotic pressure does not forbid water to pass through the semipermeable membrane, but it creates a compensatory flux of water in the opposite way. Can solutions of the same concentration of different solutes have different osmotic pressure? The osmotic pressure of a solution does not depend on the nature of the solute. It depends only on the quantity of the molecules or particles in relation to the total solution volume. Solution with the same concentration of particles, even containing different solute, exert the same osmotic pressure. Even when the solution contains a mixture of different solutes, its osmotic pressure depends only on its total particle concentration, regardless of the nature of the solutes. How are solutions classified according to their comparative tonicity? Comparative to another, a solution can be hypotonic or hypoosmotic, isotonic, isosmotic, hypertonic, hyperosmotic. When a solution is less concentrated than another, the adjective hypotonic is given. The more concentrated is called hypertonic. When the two compared solutions have the same concentration, both receive the adjective isotonic. So this classification makes sense only for comparison of solutions. Concerning permeability, what type of membrane is the cell membrane? The cell membrane is a selectively permeable membrane. That is, it allows the passage of water and some selected solutes. What are the basic constituents of the cell membrane? The cell membrane is formed of lipids, proteins, and carbohydrates. Membrane lipids are phospholipids, a special type of lipids to which one extremity, a phosphate group, is bound, thus assigning electrical charge to this region of the molecule. Since phospholipids have one electrically charged extremity along the neutral organic chain, they can organize themselves in two layers of associated molecules. The hydrophilic portion, which is polar, of each layer faces outwards in contact with water. A polar molecule too of the extracellular and intracellular space. And the hydrophobic chain, which is nonpolar, faces inward, isolated from the water. Because this type of membrane is made up of two phospholipid layers, it is also called a bilipid membrane. Membrane proteins are embedded and dispersed in the compound bilipid structure. Carbohydrates appear on the outer surface of the membrane associated with some of those proteins under the form of glycoproteins or bound to the phospholipid forming glycolipids. The membrane carbohydrates form glycocalyx of the membrane. This description with further explanation is known as a fluid mosaic model about the structure of the cell membrane. What are the respective functions of phospholipid, proteins, and carbohydrates of the cell membrane. Membrane phospholipids have structure function. They form the bilipid membrane that constitute the cell membrane itself. Membrane proteins have several specialized functions. Some of them are channels for substances to pass through the membrane. Others are receptors and signalers of information. Others are enzymes. Others are cell identifier, cell labels. And there are still those that participate in adhesion, complex between cells and between internal surface of the membrane and the cytoskeleton. Membrane carbohydrates associated to the protein or to the lipids are found on the outer surface of the cell membrane, and they have a genolabeling function for recognition of the cell by other cells and substances. For example, they differentiate red blood cells in relation to ABO blood group system, immune modulation functions, pathogen sensitization functions. What are differentiation of cell membrane? In some types of cells, the cell membrane represents differentiation that are necessary for specific functions of the cells. The main differentiations are the microvilli and the structures for reinforcement of adhesion or union between cells, that is, cell junctions. Microvilli are multiple external projections of the membrane, resembling glove fingers. This differentiation is found in the cell of tissues, where it is advantageous to increase the size of the surface area in contact with the exterior. For example, in enteric or intestinal epithelium, for absorption of nutrients. Membrane differentiation for reinforcement of adhesion between cells occur mainly in epithelial tissues, where the need for convergence, coverage, and impermeability require cells to be glued to the neighboring cells. This differentiation can be interdigitation, desmosmos, tight junctions, zonula occludens, zonula adherens, adherens junction, and gap junctions. What is the relationship between concentration gradient and active and passive transport? Passive transport is the movement of substances across the membrane in favor of their concentration gradient, for example, from a more concentrated region to a less concentrated region. Active transport, on the other hand, is the transport of substances across the membrane against their concentration gradient, from a less concentrated to a more concentrated region. In passive transport, because it is spontaneous, there is no energy spent. The active transport, however, requires energy. Active transport works to maintain or to increase the concentration gradient of a substance between two regions, while passive transport acts in a manner to reduce the concentration gradient. What are the three main types of passive transport? The three main types of passive transport are simple diffusion, osmosis, and facilitated diffusion. What is the energy source used in active transport through biological membranes? The energy necessary for active transport against the concentration gradient of the transported substance to occur comes from ATP molecules. Active transportation uses chemical energy from ATP. What is the difference between simple and facilitated diffusion? Facilitated by which type of molecules does the term facilitated means? Simple diffusion is the direct passage of substances across the membrane in favor of their concentration gradient. In facilitated diffusion, the movement of substances is also in favor of their concentration gradient, but the substances move bound to specific molecules that act as permeabilizers, that is, facilitators of their passage through the membrane. How does the intensity of simple diffusion vary in relation to the concentration gradient of the moved substance? The higher the concentration gradient of the substance, the more intense its simple diffusion will be. If the concentration gradient diminishes, the intensity of simple diffusion diminishes too. How does the intensity of facilitated diffusion vary in relation to the concentration of moved substances moved? What is the limiting factor? Like simple diffusion, facilitated diffusion is more intense when the concentration gradient of the substance increases, less intense when the gradient lessens. In facilitated diffusion, however, there is a limiting factor: the quantity of the permeases that facilitate the transport through the membrane. Even in a situation in which the concentration gradient of the diffusing substances increases, if there are not enough permeases to perform the transport, there will be no increase in the intensity of diffusion. This situation is called saturation of the transport proteins, and it represents the point at which the maximum transport capacity of the substance across the membrane is achieved. Without saturation of transport proteins under the same concentration gradient, how can the speed of simple diffusion be compared to the speed of facilitated diffusion? The action of facilitator proteins in facilitated diffusion makes this type of diffusion faster than simple diffusion under equal concentration gradient of the moved substances. How does facilitated diffusion present similarities with enzymatic chemical reactions? One of the main examples of facilitated transport is the entrance of glucose from blood into cells. Glucose from blood binds to specific permeases, hexose transporting permeases, present in the cell membrane, and by diffusion facilitated by these proteins, it enters the cell to play its metabolic function. Facilitated diffusion resembles chemical catalysis because the transport substance binds to permeases like substrates bind to enzymes. In addition, after one transport job is concluded, the permease is not consumed and can perform other successive transports. What are some examples of biological activities in osmosis, which osmosis plays an important role? Hemolysis, destruction of the red blood cells by entrance of water. Hydric regulation in plants. The entrance of water in the xylem of the vesicular plant are all examples of biological phenomena caused by osmosis. Excessive dilution of the blood plasma causes by osmosis the entrance of too much water into the red blood cell and then destruction of these cells, hemolysis. Osmosis also is the main process of maintenance the flaccidity, turgid, and plasmolytic states of the plant cells. Osmosis is one of the forces responsible for the entrance of water into the plant roots, since root cells are hypertonic in comparison to the soil. What do facilitated diffusion and active transport have in common? What are the differences between them? Facilitated diffusion can be confused with active transport because both processes there is participation of membrane proteins. In active transport, however, the transported substance moves against its concentration gradient with energy spent. Facilitated diffusion is passive transport in favor of the concentration gradient and does not require energy. Which of the molecules that make possible active transport through the membrane? Active transport is made by specific membrane proteins. These proteins are called pumps because they pump the moving substance through the membrane using energy from ATP molecules. How does sodium-potassium pump present in the cell membrane work? What is the importance of this protein for the cell? Sodium-potassium pump is the transport protein that maintains the concentration gradient of these ions between the intra and extra spaces. This protein is phosphorylated in each pumping cycle, and then it pumps 3 sodium ions outside of the cell and pumps 2 potassium ions inwards. The phosphorylation is made by binding a phosphate donated by one ATP molecule that then is converted into ADP, adenosine diphosphate. The job of sodium-potassium pump, also known as sodium-potassium ATPase, is fundamental to keep the characteristic negative electrical charge in the intrasite of the membrane of the resting cell and to create adequate condition of sodium and potassium concentration inside and outside the cell to maintain the cellular metabolism. What is mass transportation across the cell membrane? Mass transportation is the entrance and exit of substances in and from the cell engulfed by portion of the membrane. The fusion of internal substances containing membranous vesicles with the cell membrane is called exocytosis. The entrance of substances into the cell after they have been engulfed by the projection of the membrane is called endocytosis. What are the two main types of endocytosis? Endocytosis is the entrance of material in the cell engulfed by the portion of the cell membrane. Endocytosis can be classified into pinocytosis or phagocytosis. In pinocytosis, small particles on the external surface of the membrane stimulate the invagination of the membrane inward, and vesicles full of that particle then detach from the membrane and enter the cytoplasm. In phagocytosis, bigger particles on the external surface of the membrane induce the projection of the pseudopodia outward, enclosing the particles. The vesicle then detaches from the membrane and enters into the cytoplasm, receiving the same name, phagosome. How does the plant cell react when it is placed under hypotonic media? The plant cell wall, the covering of the cell external to the cell membrane, is made of cellulose, a polymer of glucose. When the cell is put under hypotonic medium, it absorbs too much water through osmosis. In that situation, the cell wall pressure acts to compensate the osmotic pressure, thus forbidding the excessive increase of the cellular volume and the cell lysis. What is meant by suction force of the plant cell? Does the suction force facilitate or make difficult the entrance of water into the cell? The suction force is the osmotic pressure of the plant cell vacuole of the vesicular internal solution. Since the vesicular solution is hypertonic in comparison to the cytosol, it attracts water, thus increasing the cytosol concentration. With the osmotic action of the vacuole, the cytosol becomes hypertonic in relation to the exterior, and more water enters the cell. What is the wall resistance of the plant cell? Does this resistance facilitate or makes difficult the entrance of water into the cell? Wall resistance or tag pressure, TP, is the pressure made by the distension of the plant cell wall in opposition to the increase of the cell volume. The wall resistance works against the entrance of water in the cell. That is, it acts forcing the exiting of water and compensating the entrance of the solvent by osmosis. What does the formula dPd equals to SF minus Tp mean? dPd is an abbreviation for diffusion pressure deficit. SF, suction force, is a vacuolar osmotic pressure, and Tp is the tag pressure. The difference between the suction force and the tag pressure determines whether water tends to or not to enter the cell. If the suction pressure is greater than the tag pressure, that means that the diffusion dPd is greater than 0, and water tends to enter the cell by osmosis. If Tp is greater than SF, then dPd is less than 0, and water cannot enter the cell by osmosis. What are the values of dPd for plant cells under hypertonic, isotonic, hypotonic medium? In plant cell under hypotonic medium, there is loss of water for the exterior, meaning that the suction pressure is greater than 0. The vesicular pressure is high because it is concentrated. TPO is equal to 0. There is no distension of the cell wall, since the cellular volume is reduced. So dPd is equal to SF. This cell is called plasmolysed cell, situation characterized by the retraction of the cell membrane that detach from the cell wall. In the plant cell under isotonic medium, there is no increase in internal water volume. SO4 is greater than 0. Tp is equal to 0, since the cell wall is not distended. The cell membrane slightly touches the cell wall, and in this situation, the cell is called flaccid cell. In plant cells under hypotonic medium, there is a tendency of the water to enter, meaning SF is equal to Tp, since osmotic pressure is totally compensated by distension of the cell wall, and dPd is equal to 0. The cell that has expanded itself to this point is called a target cell. What is the formula for dPd for withered shrunken plant cell? How is this situation possible? Withered plant cells are those that have shrunk due to loss of water by evaporation without enough replacement. In this situation, the cell membrane retracts and detaches from the cell wall. The cell membrane, moreover, expands in length to stimulate the entrance of water, making tag pressure greater than 0, less than 0, dPd equal to SF minus Tp, and Tp is negative. Formula becomes dPd is equal to SF plus Tp. What is deplasmolysis of plant cell? The plant cell, when placed under hypertonic medium, loses great amount of water and its cell membrane detaches from the cell wall. In that situation, the cell is called plasmolysis, plasmolysed cell. When a plasmolysed cell is placed under hypotonic medium, it absorbs water and becomes target cell. This phenomenon is called deplasmolysis. Why are salt and sugar used in the production of dried meat and dried fruits? Substances that maintain high hypertonic environment like sugar and salt are used in the production of dried meat, fruits, or fish. Because the material to be conserved is then dehydrated and the resulting dryness prevents the growth of the population of decomposer beings, since these beings also lose water and die. Thank you very much for participating in cell membrane. Please do like, share, and subscribe.